friends, so today I'm going to teach you the rules for the inheritance of traits. So before we get into the topic, let's see about its definition. It is the transmission of a gene from parent to child. And I don't think that you all may find it difficult because everyone knows that the child's face or the structure or its body structure is always similar to one of its parents, right? What is the reason? The reason is, of course, of inheritance. So before we get into any more details about this inheritance topic, let's see about some of the examples that we may find in our daily lives. See, in here I have drawn the images of fathers, mothers, and the child's earlobes. So what is earlobe? See, earlobe is the hanging portion from an ear or sometimes rather attached. It is the portion which doesn't have any cartilage, so it's a really soft tissue. See here, the father's and the child's earlobe. You can see here carefully, they both are looking same, while the mother's earlobe is big different. What is the reason? The father's and the child's earlobes are hanging outwards. You can see here, they are not attached to the skin, while the mother's is attached to the skin. Then in here, I have introduced two more names here. One is the attached earlobe and the other one is the detached earlobe. Okay? So why did the detached earlobe get inherited to the child? Or why did it get transferred to the child itself? The reason is that this detached earlobe is a dominant trait and this mother's attached earlobe is a recessive trait. So you may think, what are these names? I haven't heard of them anywhere. Then I will explain. Dominant trait is that trait which is inherited to the next generation or expressed, while the mothers, that is this recessive traits are not expressed in the next generation. But when we get on further, we get to see that this recessive trait will also be expressed in the next generation, but not on this first generation, but rather the next generation that occurs after this child. So now let's clearly get into the dominant and the recessive traits. So as I've mentioned earlier, dominant trait always gets expressed in the next generation. So we always denote dominant trait in capital letters, okay? We denote it always with a capital letter. We always denote it with a capital letter. And I told that it gets expressed in the first generation. So the first generation, we always mention it here as F1 generation. We mention it as F1 generation. And now we talked about the next generation where the recessive trait might also get expressed. And that generation we consider as F2 generation. So now let's also see about the recessive trait. Recessive trait, it is always denoted by a small letter, okay? For a small letter, we can use any of the alphabet's letter for A, B, D, L, M, N, O, P, anything. So, when recessive trait get expressed in the F2 generation or even afterwards, then dominant trait, it gets expressed in the F1 generation and also it gets expressed in the next generation that comes following. So, now we will see the next process that determines the rules for inheritance that Mendeleev contributed in his life. So now I'm going to tell you the contrasting characters Mendeleev took for this experimenting his progeny relationship with their parental generation. See, the first is he took round and wrinkled seeds. Means you all know some plants produce round seeds and other wrinkled seeds. So this was one basic contrasting character. The other one is tall and short plant the other is white and violet flowers. See, before we get more details about this portion, I'm going to tell you these all characteristics were seen in one plant, that was pea plant. So in many questions in NCERT textbook or in sometimes in exams, they may ask that why pea plant was selected by Mendeleev to perform this experiment. So the main points that I can highlight is here that pea plant is a small plant. You all know this, right? Pea plant is a small plant, and further if we see that it is easier to grow in gardens, which means it doesn't require much more maintenance and or much more water. And also the third point is that lots of contrasting characters or characteristics is seen here. As I have mentioned earlier, that round, wrinkle, tall, short, white violet, these are a large amount of contrasting characteristics. You may not find these contrasting characteristics anywhere in other plants like mango. It may occur, but it's a really big plant, right? So we easily find pea plant and we can grow it easily. So this is the main plant we find it easy to perform experiments. So that is what Mendeleev also taught. So let's get on to the experiment portion. Mendeleev took at first to compare tall and short pea plants. Okay, tall and short pea plants. This is the main commonly found noticeable difference in a pea plant. So first he took this pea plant, and I'm just drawing pea plant. If it's wrong, don't mind. Okay. See, I am denoting capital T. I have told that dominant traits are always capitalized. See, then T I have kept here capital, and T is for tallness. T is for tallness, and I have taken another pea plant which is short. See, this is a recessive trait. Okay, so we have taken here smaller t, means this signifies shortness trait. So when we try to get progeny out of them, means when we try to cross-pollinate them, you all know what cross-pollination is, right? When it's transfer of stamen or stamen to the ovary of other plant, then this is called cross-pollination. So when we try to get progeny out of them, we get a tall plant. You might already have guessed the reason why I told that we would get tall plant. I've told that always dominant trait is been expressed in the F1 generation. Now let's see into the zone portion, okay? In here, we don't write a single T. We write double T. Okay? Means to signify that it too had two parents. Okay? And this too had two parents. Then we will write this as T. See here. First Mendeleev took the tall pea plant and he cross-pollinated it with the short pea plant. Right? Oh, I forgot to ask. You all know what cross-pollination is, right? It means two different plants that have different characteristics are used to produce a progeny from them, or they both perform reproduction to make any progeny. And self-pollination means the same plant or same kind of plant is used to produce a progeny. Okay, then in here, this is the tall pea plant and this is the short pea plant. Then when he crossed, you have to listen to this very carefully. See, I'm going to show you the genetic transfer. Okay, so it should be clear. See, this T, the arrow mark should be given like this. And this would go with this T also. And this T will go with this T. And this T will go on this T. In here, This is capital T. Okay, from here I got capital T, and to which arrow mark I have pointed? Okay, this T. Then this is small t, and here this capital T, this small t, and respectively we got all this TT. Okay. Then here I told you that tallness is a dominant trait and shortness is a recessive trait. So this is the F1 generation. Okay. And I have told you earlier that the dominant trait is to be expressed in the F1 generation, while recessive trait can be found out in the next generation, means F2 generation. So now this all would be tall, because T comes from the dominant trait, and this small t comes from the recessive trait. So I hope that it's very clear to you right now. So let's recall which progeny Mendeleev obtained by crossing them. Okay, the first one was TT, and the next one was TT, next one was TT, and this two also TT. Okay, then what did Mendeleev do in the second step? He self-pollinated these two same characteristics type. Okay, means he self-pollinated TT with this TT. Now you may have guessed what will happen in the next process. Okay, see, we pollinated them, and what happened? This arrow will go to this arrow will go to this arrow, and this to this, and this to this. So now you have to look carefully that how I'm writing the combinations. Here this T is going to this T, then it will come TT, in this T to this T, then TT, and this T to this T we get a TT, while this T to this T will get a TT. Then in here you have seen that in here there is no dominant trait found. Okay, only recessive traits are being present here, while in these trait types there is the presence of dominant trait. Then this tree would have tallness, and this would have the shortness characteristics. Okay, now it's clear, right? Recessive trait would be expressed in the next generation, means F2 generation, only if two recessive traits of the same type are found there. It's easy, no? And this dominant trait, whether it is found in the two genes or the single, then too it will express itself in the progeny. It's similar to the one that I showed in the earlobe example. Okay, in this experiment we found out that there were no medium-sized plants. Okay, there were no medium-sized plants, means between tall and between short. The things that were expressed were tallness or the shortness trait. There were no something between them. That is the reason that this trait is called independently inherited trait. So I will just represent this by using a diagram. See, I am using this line as tallness. Okay, so this is tallness, means tall plant, and we have crossed it with short plant, and this is shortness, this small line. Okay, so what did we get it as its progeny? We got a tall plant which was same to that of this tallness, and this happened in the F1 generation, and in the F2 generation we also got the tall plant and the short plant. But did we get something medium sized or something that's between these two, shortness and the tallness, means rather this size? No, we didn't get it, right? We only got the tallness or the shortness if we compare it with both the F1 and the F2 generation. And this this makes us the other rule that the tallness and the shortness are independently inherited traits. So there will be no other trait that will be mixed by these two, or rather combined. Now let's take other experiment that Mendel carried out to find out that some new mixtures came to the existing progeny. So this doesn't say that they were independently inherited traits that were crossed. So I'm going to give an example. The first characteristics we are going to take is tallness. round seeds. This is the first plant. And the second plant, which is to be cross-pollinated with this plant, is having the shortness trait and wrinkle seeds. In here, I have taken tallness and round seeds and shortness and wrinkle seeds. I have taken two characteristics in each of the plant. In here, if you like to use the arrow method, but we can't use it here definitely, so we will just simplify it. For here, we are using one single T and one single R, and one single T and one single R from here. So when we crossed these two from genetic transfer, then we will get T R T R, or rather we can write T T and R R. And here you might have noticed that this T is dominant, so it will have tallness at first. And the other characteristics, this R, this is dominant, so we put it capital. So the dominant trait would be round. So tallness and round is the dominant trait, and its progeny also will get the same trait. Okay. So now let's see which progeny we may obtain in the F2 generation if we cross the progeny we got in F1 generation. Okay. Then let's see. So for that we might have to see which progeny we got in the F1 generation. We will just recall them, TTRR and these two TTRR. And when we pollinate them, when we self-pollinate them, then we will get to find out that new mixtures are being obtained in this progeny. So for showing the new mixtures, it's rather easy to learn it in the table way like it is shown in the book. Because for simplifying, we will have to write TR, 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 TR. Similarly, we have to write the simplifying method of this, and we have to crossbreed each of them with everyone. So it is really a big number that we're going to get after this. So we'll just see the table method, okay? So now I'm going to show you the table method. So how to cross in a table method. So our progenies were TTRR crossing with TTRR. So the table is really easy to draw. First we have to draw a rectangle, and we have to draw like this, rows, and now we will simplify it. This is TR and TR, and this is TR and TR, and this is similar to this thing. So we will write like this, TR, TR, TR and TR. Similarly, we will copy it here. Now we have to draw more rows and columns. So we will draw them. Now see, now we have to multiply them. You know in maths we do it TR, TR, then it will be TTRR, and similarly this will be TTRR, and this will be TTRR, and similarly we can write all the progenies. See, now I have filled out this table. Now we can see that here the dominant traits are coming together, all dominant. So it's true that it will be tall and round seed. And see here it is full recessive traits, so it will be short, and wrinkle seeds. So in this way we can make and calculate the progenies that is coming from the F2 generation after crossing these two progenies that we obtained in the first generation. And here we are showing that while we are crossing them, there are certain circumstances when these recessive traits are also being expressed in this generation with addition to the dominant trait. So now we are trying to show that in here all things are not independently inherited. We can see here, see this is TTRR, and what is TT? It is short. And what is RR? It means round.
So a new mixture has been seen here, short and round seed. And similarly we can say here, it is tall and wrinkle seed. These are the new mixtures that we get to find in this progeny. So in here, the traits are not independently inherited. So I hope that this video helped you to learn the rules for the inheritance of traits, right? So if you like this video, then please don't forget to like it and subscribe it. And also you can click on the notifications bell icon so that you can get the further videos that we upload in this channel. So thank you.